Hi, my name is Raman. I'm an assistant professor in the Department of Chemical and Biological Engineering. I also have appointments in the biochemistry department, which is where my lab is located, and the bacteriology department. My lab works in the area of systems and synthetic biology. The overall goal of my lab is to, one, understand how biomolecular systems work, and two, to engineer biomolecular systems with new properties. And to accomplish this goal, we systematically perturb biomolecular systems through high-throughput mutagenesis and measure their function. And so from these studies, we will get a sense of what the overall functional landscape of the biomolecular system looks like. And using this information, we can now engineer these systems to do cool new things. And so these mutational perturbations can be both gene variants, where we systematically mutate a single gene, or could be genome variants, where we do systematic knockdowns of a gene, of every gene in an organism. And just to put a number, the scale at which we do these experiments is thousands to tens of thousands of mutants per gene or genome. Now, these are obviously not done one at a time. We develop what are called as pooled experiments, where all these mutants are assayed in a single pot experiment. And so now the question is, how do we measure function? We take a variety of approaches to measuring function. The function of the biomolecule could be directly coupled to the function of a reporter. The function of the biomolecule could be coupled to cellular fitness, or the function of the biomolecule could be coupled to single-cell RNA sequencing, where we then map each mutant to the entire transcriptome of the cell. And using these principles, we apply them to three application areas. First is engineering allosteric biosensors. Second is engineering bacteriophages or phage bacterial interactions. And third is mapping the effect of mutations in human disease genes. So allosteric biosensors are biomolecules that detect specific small molecules or metabolites in the environment. And so these biosensors are used for medical diagnosis, for environmental monitoring, for bioremediation, and bioenergy. And so the key challenge here is not just to sense, but also to activate a signal in response to sensing the small molecule. And so therefore we use allosteric transcription factors as biosensors, because upon sensing the small molecule, they can turn on gene expression either of a cell-based system or a cell-free system. Our second goal is to engineer bacteriophages, and bacteriophages are viruses that infect bacteria. And so we want to understand what the interaction landscape is between phages and bacteria, and to engineer synthetic phages for the treatment of drug-resistant bacteria, to kill drug-resistant bacteria, and as a tool for precision microbiome editing. Our third goal is to map the effect of mutations in human disease genes. And so we use massively parallel reporter assays and single-cell RNA sequencing to map mutations in human nuclear hormone receptors. And so these human nuclear hormone receptors are among the top drug candidates for human diseases. And the goal here is to advance precision medicine by classifying mutations into different subgroups, and so that we can then precisely tailor drug screens to different subtypes. So my lab uses a combination of both computational work and experimental work. And so we use the Rosetta software for computational protein modeling. And we use and develop high-throughput experimental methods for genome engineering, DNA synthesis, and sequencing technologies to measure function. So my lab is primarily run by graduate students from a variety of different programs. And I invite you to visit the lab website for more information.